Welcome, everyone. It is officially 1 o'clock and time to kick off the afternoon session of day 3 of Washington Soil Health Week, SoilCon. I would like to offer you all the friendly reminder to use Google Chrome for optimum viewing of the presentations. If you have technical difficulties, you can use the support chat widget located at the bottom left of the screen. Alternatively, the chat bar is where you can submit questions for the presenters. So we will have a discussion at the end of this at the end of the viewing of the video recordings today. And at the end of the day today, you will be prompted to answer a quick poll to survey you about your experience. This is valuable information to continue programming like this in the future. So without further ado, I would like to kick off hearing from Buzz Clute. Dr. Clute is a research associate professor in the Environmental Health Sciences Department at the University of South Carolina. He will be discussing biological soil health indicators and practical end uses. So let's kick off that presentation.
Hey everybody, it's Buzz Clute here. You've seen my bio and you've seen my picture, and I think you've probably seen my video over here. So we're really trying to look at biological soil test indicators and then compare them to chemical soil tests and really use them as a kind of an important and imperfect window into what's happening in the soil. And we're taking different soil tests from four different types of soil tests from different labs, and we looked at 3 different management types of soil. We looked at degraded soil, soils that are in progress of getting better, and then regenerative soils. And in our part of the world, we've got the Piedmont and the coastal plain, and so we looked at 2 different ecosystems. And I'll try and give you a few thoughts and observations on that. The first soil that we looked at was, this is a soil in my backyard, and I don't know if there's anything that is more fungal that I've ever seen in any other soil. So it's been under mulch or under multi-species cover crop. I grow roses in it, and then we also have turmeric plants that we grow and then sort of feed ourselves for about 3 to 5 months of the year with turmeric from this little garden. And it's a terrific garden, very, very good soil structure. You know, you can actually see there's a terrific amount of soil organic matter in it as well. And it started out about 10 years as very degraded, but this is a pretty decent soil. Over here, this is a pasture. One of our farmers that we work with, and it used to be cotton, degraded cotton, and we have the farmer turned it into a pasture, and we're throwing multi-species cover crops into that as well, as we want to turn it into a perennial native warm season pasture as well. It's a sisal soil, and the surface is probably it's no longer really an A horizon, it's a B horizon. The top 1 meter over the last 100 years has been washed off. So we have an A horizon of maybe, 3 inches or so. And we have a very degraded coastal plain soil. It's what we call a Norfolk soil. And it's been tilled and fertilized and chemicalized to death in the last, 30 to 40 years. Often sort of the crop rotation there was collards and soybeans, so very little grass. And actually what you see is our attempts to start growing a cover crop on that. In terms of the soil tests that we tried out, we looked at 3 labs. One was Cornell. Cornell does their own soil test suite, and they gave us they give you the chemistry, Morgan and modified Morgan. They look at aggregate stability. They look at soil carbon as well. being, let's have a look, being both organic matter, and they're looking at a 96-hour carbon dioxide test, and then they're also looking at active carbon, and then they're looking at soil nitrogen with autoclave citrate or extractable or ACE soil proteins. We also get a standard soil test from our land-grant university, Clemson, Malik 1. It's one of the few universities, I think Georgia also does Malik 1 extraction as opposed to Malik 3 anymore. But we get, we then also ask them to do organic matter, and they can actually give nitrate, soil nitrate in. We went to Ward Lab that gives us essentially the H3A test. What we call Haney and Hosner, that gives us the soil. So it also gives us organic matter, but it gives us water extractable organic carbon, the 24-hour CO2C, the Haney-Brinton test. And then they look at water extractable organic nitrogen. They look at ammonium nitrogen and nitrate nitrogen. So they've got both the inorganic and organic nitrogen. And then Ward Lab also does a phospholipid fatty acid test, which basically gives biomass, and mainly bacteria, fungal, and then some of the protozoans, or basically predators. And then one of the local commercial labs that we have, Waters Lab, they do both Malik 1 and 3 extractants, and they've added a soil health test to their suite of things. So they do aggregate stability. I think it comes from the Solvita method. Organic matter, they do the 24-hour CO2C method as well. They use the Solvita paddles. And then they also do ammonium nitrates, and then they do what is called the Solvita labile organic nitrogen, or SLAN test. So those are the tests that we're looking at over here. And let's go ahead and see what goes on. So in terms of soil chemistry, what you have over here is the 3 soils that we sampled. So this is my rose garden, this is the pasture, and this was the coastal plain soil. And in my rose, or my turmeric garden, what was interesting, I guess what's interesting in the soil test P, is the ranks are pretty much the same. You can see, regardless of what extraction system we used, you know, this was the highest, this was the lowest, and then this was somewhere in between. What was real interesting in this rose garden is that I haven't added, ever, I haven't put a lick of phosphorus fertilizer or even manure onto that land. It was all basically wood chips and multi-species cover crop. The pasture over here has a very, very high clay content, and so you can see, you know, very little of the phosphorus actually shows up even when we try and extract it. And then in the coastal plain soils, we see it's sandy, and typically in our part of the world, the sandy soils usually turn up with more phosphorus in that. If you go to the soil test potassium, you'll see again, not surprisingly, this really, really good soil is very, very high in potassium. Again, I've never added potassium to the soil. And then in the coastal plain soil, because it has high clays, it tends to have a slightly higher CEC, and you can see some of those numbers over there. And then in the degraded coastal plain soils, you'll see much lower potassium levels, again, because of a low CEC. So interesting, you know, it doesn't matter what, you know, we had the Cornell, Clemson, Ward, and Waters. Each one of these have different extracting methods. You know, if you're using the same extraction method, the outcomes, you know, as long as you're using the same methods all the time, I'm not really that worried about, the exact number for soil chemistry. I like to look at trends over time, and we'll talk about that a little bit later on. In terms of soil carbon, you'll see this is the blue bar is the organic matter for Cornell, Clemson organic matter for Ward, and this would be the Waters lab. And you can see there much of a muchness. Ward was slightly lower. If you look at the pastures, you're kind of seeing the same thing. Again, the ranks are about the same, and not much to say there. But you can see the labs are coming out. I'm not too worried about any huge difference. Once we come to carbon respiration, Ward and Waters are using the same method. So that's Ward is the dark blue, Waters is the purple. Oh, sorry, excuse me. This is Cornell over here in the dark blue, and then Ward and Waters over here, the 24-hour carbon. And you can see it's quite different. So in terms of the Cornell, the blue bars, again, you can see the ranks are the same from regenerative soils to, pretty poorly degenerated soils. And you can see those ranks are the same. What we have found in the Haney, when we do the 24-hour CO2C test, we've noticed that in Haney's lab, that they used to use the Solvita paddles. These are paddles with colorimetry, and when you have very, very high numbers, they tend to saturate out. So what Haney has is he has a Frankenbreather that measures all of the carbon dioxide that comes off. So you'll see big differences between these soil tests if you've got the Solvita paddle versus the Haney Frankenbreather, which Ward has. And then you can see that difference is a little bit smaller when you don't have that much of when you don't have that much respiration in the soil. And then actually it's slightly reversed over here when you have low amounts of respiration. In terms of the other way people look at carbon is the active carbon, the KMNO4. This is again the Cornell value. You can see the Cornell in the dark blue here. Again, we can see the ranks. In the middle, it's water extractable organic carbon. That's the Haney method that Ward uses. And again, you can see the ranks. And then finally, the active carbon, and that's what Waters uses. So again, it's apples and apples, but what we see is the rankings are about the same. And there are different ways to measure carbon. Which method is the better method to measure carbon? I can't tell you. All I can say is, again, we have to, if you're going to choose any one method, for goodness sake, be consistent rather than comparing one lab versus another. You can see our nitrates. Let's have a look. This was for Ward and Waters. Waters tended to measure a little bit higher than Ward, and who knows what that was about. It might have been that Waters got our samples a bit quicker because they're in the state next door, and Waters in Nebraska. In terms of ammonium, again, much of a muchness. We rarely see differences. We rarely see big fluctuations in ammonium. 1 to 5 or 6 parts per million is really all we tend to see. Soil organic nitrogen, again, we've got 3 ways of measuring it again. So it's apples versus apples. And again, all I want you to do is pay attention to the rankings, which tend always to be consistent. Sorry, this is apples versus oranges. So this is the ACE protein index that Cornell has, the Ward, the Ward one, which would be water extractable organic nitrogen, and then the Solvita labile ammonium nitrogen that our Waters lab does. So those are some of the ways that people look at not only the inorganic chemistry, but also some of the chemistry associated with the nitrogen and the carbon. If we start looking at soil microbes, again, the phospholipid fatty acid is not the be-all and end-all, but what we like about it is it gives our farmers a window into soil biology, and it helps them understand what the biology is. This is a picture I took on my high school microscope over here. I don't have very sophisticated equipment, and this is a lovely strand of fungus on one of the soils, and I think that was from my rose garden area. So in terms of the PLFA, our total biomass, PLFA biomass, so that's nanograms per gram. So that's the amount of phospholipid fatty acids are usually found in the cell walls of these microbes, and they can distinguish between bacteria, fungi, and then it also looks like the protozoans. And you can see again this ranking. So really highly regenerated soils, 3 times better than the one that's getting better over time, versus this little guy over here where we find not very much life. So we can see that the bacteria and then the fungal biomass. This is quite a nice fungal biomass over here, especially for our part of the world. So in terms of fungi, you know, we've got the saprophytes over here, and then we also have mycorrhizal fungi that live in symbiosis with the plant roots. This is a corn root hair, and we were fortunate enough to get this symbiotic fungus growing there. So, you know, basically these guys actually are able to distinguish between saprophytic fungi and then also the arbuscular mycorrhizal fungi. So that also helps distinguish a little bit between what kind of fungi we have in the soil. This is a testate amoeba, and these little dots around here are typically bacteria, and the testate amoeba basically has a test or a shell around it, and it munches these things. So it's just basically a fertilizer bag opener and spreader, because bacteria are fertilizer bags, and this testate amoeba is just going to digest them, and it's going to poop and pee out nutrients right next to the rhizosphere. So what we find is protozoans, or what they talk about predators. We've got a lot of predators in that rose garden area, quite a few in the pasture, and none in this degraded area, possibly because they've been applying all sorts of pesticides for a long time. And you can also see significantly different fungal to bacterial ratios, and we typically look at fungal to bacterial ratios as ways of measuring soil health. So is there only one way to interpret a soil health test? I think people just often kind of get stuck on, well, what does the soil test say, without really understanding what happens. And I love this picture, Dr. Brad Jorn, in a presentation not that many years ago, basically started to look at the fertilizer costs of 180 bushel corn and 60 bushel soybeans over 2 years, rotation over 2 years, and started looking at the annual costs based on land-grant university fertilizer recommendations. Often people criticize something like Haney and say, well, the Haney stuff hasn't been calibrated But in reality, what we find are enormous differences between different land-grant universities that are right next to each other. And Dr. Jones says, well, you know, what happens if you're in Indiana and you've got 1,000 acres over here and another 1,000 acres in Illinois? You know, in Indiana, your costs are going to be $235,000 a year. In Illinois, they're going to be $392,000. Same thing with Georgia and Alabama. If you've got a farm on the border, which are you going to choose? So we tend to have this impression that our fertilizer recommendations are set in stone, but in reality, a lot of these are built on tradition and pride. As Dr. Jones says, these are not my words. And, you know, there is science that goes into it, but interpreting that science, you know, there's a lot of interpretation that goes to it. A lot of these things have been done on things like corn and soybeans and wheat and other staple crops, but they also seem to be extrapolated. In my own home state, you know, there have been a lot of extrapolated values that have come in as well over time. And I'm not blaming people, but I'm just saying this is what it is. So the soil test, you know, who's doing the soil test, who's been doing the calibrations? Were the calibrations done on no-till soils? Were they done 50, 60 years ago when the soils were tilled up? So, you know, what were those conditions? And so I think this whole area of fertilizer recommendations needs a lot of rethinking. And certainly, you know, in the little work we've done, we have found that a lot of our kind of results have to do with potassium and nitrogen have really subverted some of the long-held beliefs of the necessity of lots and lots of potassium and nitrogen in sandy soils over here. So a lot of people also, the Haney test is like, there's people say, well, what's the Haney test, you know, and people are often thinking about a number. And so I wanted to sort of borrow a few words from Rick Haney and Ray Ward, who talk about this a lot. There are three parts to the Haney test. The first part is the 24-hour carbon dioxide test. So it's a respiration test to measure microbial biomass. It's an indirect measurement. 60 to 150 parts per million is pretty good, but it could be as high as 800 parts per million. So if you're using that little Solvita paddle, you know, you're saturating out at about 100 parts per million. And as we get better and better and more regenerative soils, you know, our ways of measuring are getting better and better. So, Ward labs and the Haney labs are doing that with their so-called Frankenbreather. There's also a water extract for measuring carbon and nitrogen in soil solution. So they basically take a known amount of water and a known amount of soil, and they do a simple water extract. As Rick Haney says, when you shower, it doesn't rain, you know, malic 1 or sulfuric acid and nitric acid or whatever, or any of these other solutions. So they do a water extractable situation where they look at both carbon and nitrogen. So they call it WEOC and WEON. A WEOC, water extractable organic carbon, is food for microbes. Typically 100 to 300 parts per million is a good number. And a rule of thumb is it's about 80 times as much as the carbon that you'll find in the organic matter. Typically, the water extractable organic nitrogen, you know, we're finding anywhere between 15 and 40 parts per million of organic nitrogen, and 40 to 60% of it should be organic nitrogen compared to your inorganics being nitrate and ammonium. But if you're ignoring that organic nitrogen source, you're ignoring a great resource on being able to cut back. The H3A extract really does mimic plant uptake of plant nutrients. So it's a mix of malic, oxalic, and citric acids, and then they extract pK and other minerals. Again, it's just another extract that comes out, and that's the Haney test sort of in a nutshell. One of the alternative uses that I use for soil tests, again, I'm not that confident that chemical soil tests, or I have a maxim that says all soil tests are wrong, but some are useful. And I'm not always that confident that, you know, a chemical soil test is going to give you the precise amount of fertilizer that you need, especially, you know, there are lots of soil tests that absolutely ignore the amount of organic matter or nitrogen in the soil. They'll give you, they'll ask you for your yield, and it'll ignore anything else. But one of the ways that we like to use our PLFAs is for several years, we just sprayed out in this cover crop, we sprayed out for the growing period of the cover crop, we sprayed out a 20 foot by 20 foot area, and we did that 5 times across different fields of this guy, Jason Carter's place. And we wanted to see what phospholipid fatty acids did when we grew a cover crop versus killing a cover crop. And in general, what you'll find, these are significant differences, is we're finding, you know, 1.5 times more biomass for things like bacterial biomass and rhizobia, and actually double the fungal biomass. And then, of course, you know, your fungal to bacterial ratio also increases. So that was a very simple way for us to say, are cover crops actually contributing to soil biology in our soils over here? The other alternative method I've been using our land-grant University Clemson for many, many years. And these are data from some of our cover crop lands over from, it was November 13 to December 18. We had a grant from NRCS. Soil pH is a big problem over here. And when we started to include cover crops in the rotation, so we never have any fallow periods, what we noticed was we no longer had to apply lime anymore. I know some of you guys out in the West don't have that problem at all, but we noticed this. And so, you know, you can track your soil pH over time, which is great. We've also tracked our organic matter over time. And a rule of thumb for us over the last 7 years is that if we're going no-till and add cover crops and do nothing else, we're tending to add about 0.05% of organic matter per year into the system, and I think that's about 1,000 pounds per acre, if I'm not mistaken. So I want to give you a last word. You know, I hope this has just given you a very small window into our soil testing systems. You know, and I would say decide what you want to observe. You know, do you want to look at the chemical, physical parts of the soil? Do you want to, you know, physical being our soil structure? Do you want to look at the soil carbon, nitrogen, or biology? And settle in on a lab or labs that you want to use, and don't jump around, so that you've got consistent material over the years. Make sure you take your samples, you know, at the same time of the year, or same times of the year, you know, if you want to do it maybe in the spring or the fall. And just remember, there's more than one way to interpret your soil test. And like I said, my maxim is, remember, all soil tests are wrong, some are useful. So hopefully this gives you an idea of some of the different biological soil tests. And I think after this, I would be open to questions. and thank you for listening.
Great. Thank you so much, Buzz. That was a very informative talk, including that all soil tests are wrong. Some are useful. What a great nugget to take away from there. We will be holding questions for the end of the second talk from Dr. Jordan Wade, who is currently a postdoctoral researcher at the University of Illinois and will be an assistant professor of soil health at the University of Missouri later this year. He will talk about the pros and cons of soil health indicators. And just a fun tip, more information on the speakers' bios can be found at the bottom of the session page, as well as on the main website. And I definitely encourage you to go check those out, especially for Dr. Kloot. And with that, let's hear the pros and cons of soil health indicators from Dr. Jordan Wade. Thank you.
Hi, everybody. My name is Jordan Wade. I am currently a postdoc with the University of Illinois, and starting in the fall, I will be an assistant professor of soil health with the University of Missouri. Today, I'm going to be talking to you about the pros and cons of using soil health indicators. This is kind of a new-ish area, and so, there are some things to look out for, and I wanted to kind of give my perspective on that. Now, a lot of people in this conference are going to be talking about soil health and have lots of different kind of perspectives and things to bring to the table here, but I wanted to kind of zoom out and give an overview of my approach and how I see these things kind of fitting together. So obviously, when we talk about soil health, we talk a lot about management, right? There are lots of different types of management, and depending on the crop, the system, context, etc. So USDA has kind of put together these guiding principles that I think work really well as, to help with a roadmap here. So these are kind of principles like keep the soil covered, minimize disturbance, keep plants growing throughout the year, and diversify those plants. Some people also like to add incorporate livestock is another potential fifth soil health principle. Now, when we implement that management, we assume that some change in our soil will happen. We have something that we're looking for, right? And we want to make sure that that change is happening. So we use an assessment of some sort. This is where soil health indicators come in. And it's often broken down in this very familiar kind of framework where it integrates three different components of soil. So first we have the physical component of soil, we have the chemical component, and the biological component. And bringing those three things together is the goal of soil health. Now, when we're implementing some management and we are looking at the assessment, the reason is because there are lots of factors that change how the effects of management, right? So these are things that we're all familiar with, things like climate, your parent material, how long the management has been in place, and soil texture. Now, these are the reason why we can't just have some one-size-fits-all, recommendation, and why, a soil on the West Coast will be different from a Midwestern soil, will be different from one from the Southeast. So these really show that, we need these assessments, and they're a pretty important part. But this brings me to my first take-home point already. I want to emphasize that soil health indicators are not an end unto themselves. These are relatively new. As you're going to find out, there's a lot of things that we're still learning and figuring out. And so far, we don't have enough confidence in these indicators to say that this indicator reflects soil health inherently or some component of soil health. And this is not well reflected in the literature because we kind of have studies take two different forms. So a lot of initial studies take this kind of generalized form where people say, we performed X, Y, or Z good management, some soil health building practice, and this new indicator went up or down. Therefore, we assume that this indicator represents soil health, right? Management is being reflected in the assessment, therefore we're good to go. But then there's also this other form of study that happens where people use these indicators then, and they say, hey, this X, Y, or Z management that we did, that these indicators go up and down with it, therefore this management is building soil health, which, as these two kind of arrows demonstrate, we're kind of going both ways, and we're using them to justify our results. But since we're not fully confident in this assessment piece yet, this form here is a little difficult to verify. So yeah, anyways, I think that is an important thing to foreground as we move forward in our discussion here. Now, what I'm trying to say is don't stop at the assessment. What we need is this kind of next step to make sure that these indicators are telling us what we think they are. So this is where we try and connect our indicators, our assessment, to some sort of outcome, right? We want to know, what is it actually telling us? What is the information we're able to glean from that? We want to connect it to these soil health functions, things like your plant or your crop productivity, providing physical support, the nutrient cycling, the regulation of water movement, or pollutant filtration. So those types of things we would want to connect this indicator and this assessment to. Now, when we think about this, though, I think there's kind of two other take-homes that I want you to get here. So the first one is, as we are interpreting our effective management, how it's reflected in our assessment, we want to make sure that we are in fact measuring the signal from that. And I'll go into that a little bit more in depth. And the next one is that when we're using these indicators to connect to some sort of outcome, we want to be very careful with how we do that because we don't have a really good way of interpretation quite yet. So just a couple words about these indicator-based soil health assessments. As I mentioned, there are these three kind of components of soil that soil health is trying to bring together. And there are specific indicators and measurements that are associated with each of these. So physical measurements might look like aggregate stability or infiltration, compaction resistance, and water holding capacity. Similarly, chemical-based indicators would be things like permanganate oxidizable carbon, or POXC as it's called, soil protein, soil organic carbon content, potentially mineralizable N, that anaerobic test, extractable organic carbon and nitrogen, or something like an amino sugar test, think ISNT, etc. In terms of the biological ones, which is where kind of a lot of the soil health interest lies, things like enzyme activity, microbial biomass, PLFA or phospholipid fatty acid analysis, mineralizable carbon, which is also referred to as respiration, things like earthworm counts, community level physiological profiling, microbial fingerprinting and sequencing. These are all kind of biological indicators that we're working on. Now, today I wanted to focus on just a couple of these. Obviously, I'm not going to go through all of these, and this isn't even an exhaustive list. So I wanted to focus on just a couple. So I wanted to talk about permanganate oxidizable carbon, soil protein, enzyme activity, and mineralizable carbon. I think these are things that have a lot of promise for implementation, so I chose to focus on those right now. Now remember, the second take-home point that I want you to get is, first, I want you to make sure that when you're using these, that you're measuring the signal. So what do I mean by that? Well, when we implement a management and we are assessing how that management is going, there are kind of two different pieces that are coming into play, right? So we have some signal, which is the true effect of management that it is having, but then there's also some sort of measurement error, some degree of that noise, and that is also coming into play. But when we get our indicator value back from a soil test lab, both of those things are in there, and so we want to make sure to minimize this measurement error. And so really that question is, how repeatable are soil health indicators? And when we're thinking about repeatability of these indicators, what we're really trying to do is balance repeatability with high throughput. So we want to be able to take these indicators and run a lot of them very quickly in these commercial labs. We want to be able to run hundreds a day potentially. But we also want to know that even though we're running hundreds, that the values we're getting from those are reliable, right? So how repeatable are measurements? I want to kind of break that down, that repeatability piece, into two separate components. So the first one is analytical variability. This is the variability within a lab. If I'm running a lab and I run the same sample 3, 5, 10 different times, how much spread am I going to get in the data back? Another one is, can I take the same sample, send to a couple different labs, and what's the degree of agreement or disagreement between those labs? So first, I want to look at this permanganate oxidizable carbon, the epoxy. So on average, the permanganate oxidizable carbon shows around 7 to 8% analytical variability. Now, of course, there's a huge range here. We can get as low as 5, and I've seen as high as 40% within a single lab for any given sample. The variability between labs is similar, about 7 to 8%, although the range is a bit smaller. It goes a little bit lower, 3%, and doesn't go quite as high. It's around 15% variability between labs. Another popular measurement is this mineralizable carbon or respiration. And on average, that one gives us about 15% analytical variability. And again, there's a huge range going from about 5 to 60%. This one has a little bit higher between lab variability of about 20%. And again, it gets a little bit higher than the permanganate oxidizable carbon. Another new and emerging indicator is soil protein, which has pretty low analytical variability, about 3%, and that range is pretty tightly constrained, 1 to 5%. As I mentioned, this one is pretty new, and so there's not a lot of labs offering it. So we don't really have a good handle yet on this between lab variability because just not that many labs run it. Lastly, I want to focus on enzyme activities, which gives us about 10% analytical variability, again, with a large range up to about 50%. And it varies from lab to lab about plus or minus 10%. But even this is not well studied. And there are a variety of reasons for that. But I did want to kind of point out that there is, that these are roughly in order of increasing throughput. So we can run the most permanganate oxidizable carbon per samples in a day, and enzyme assays probably take the longest out of these 4. So a couple of different takeaways that I want to emphasize here. First off is that we can get the analytical variability down to pretty low, about 5% or so if you look at these, some even less. So that's possible. We need to kind of figure out internal quality assurance, quality control type stuff, and that's going to be on a lab by lab basis. But the between lab variability can still be pretty substantial. We have 7, 20%, 10%. That's a good amount of variability still. And when we think about this balance between throughput and repeatability, it's not necessarily straightforward, right? The most variable ones are pretty high throughput, and but also the most high throughput is medium variability, we'll say. So it's not necessarily an either or, for across indicators. I did want to say, just kind of give a quick summary on the currently kind of best practices that we think. So permanganate oxidizable carbon, just use a standard 2.5 grams of soil, and it's a 12-minute extraction, which is 2-minute shake followed 10 minutes of settling. And you can run that on 2-millimeter ground soil, which is what most commercial labs run their stuff on. For mineralizable carbon, the kind of best case scenario right now is to use 2 50-gram subsamples, run them together in the same kind of mesocosm, and you run them for 1 to 3 days. That incubation period is kind of up to you. We don't have a good handle on which one is best yet, but run it at 50% water-filled pore space. And again, using that 2-millimeter ground soil is good bet for you. So a protein, use 3 grams, again, on that 2-millimeter ground. And then enzyme assays, it's hard to kind of distill it down because we have so many different enzymes, CNP cycling enzymes, and lots of different substrates, et cetera. So it's a little harder to kind of distill that down into a singular recommended method, which is obviously a little tricky. So the next take-home point, which I think is probably the one that people are going to be most interested in here, is to be really careful with the interpretation of these soil health indicators when you are using them to say, hey, here's the expected outcome here. So when we are taking these indicator values and that kind of soil health assessment and using it to predict outcomes or describe outcomes, we usually use something that's called a soil health score. Now, soil health scores, calculating them is quite the journey. There's lots of pitfalls and things to look out for and obstacles here. So I just wanted to kind of highlight a couple of those and things to look out for. So when you're calculating a soil health score, there's a couple problems that we need to think about and the reason why it's so challenging. So first off, the baseline soil health varies from field to field. So what do I mean by that? We need to make sure that we are putting each soil on equal footing, right? So a soil in the arid West, right, in California and Washington is going to be very different at the start from a Midwest mollisol. These soils are very different and we need to make sure to put them on equal footing for a soil health score. And soil health is not something that we can go out and directly measure. Unlike human health, where we can just ask humans things and we have some straightforward measurements, we can't just go out and directly measure soil health using one thing, which brings me to this last piece, is we need to figure out how to integrate multiple measurements and make sense of them, make it simple and not too overly complicated when we're developing this score. So I wanted to kind of walk through the basic general steps of calculating a soil health score. I took this from the Cornell Soil Health Manual. They do a great job of explaining this. And it's a similar process for things like the SMAF, the Soil Management Assessment Framework, et cetera. But they usually use something called scoring functions. So the way to put together a scoring function is you run a bunch of samples. So here for permanganate oxidizable carbon, and you see kind of the distribution of values and what it looks like, right? So you have some that are low, some that are high, and you have a little bit more in the middle. It's kind of normally distributed here. So then you take those and you develop what's called a cumulative distribution function, right? So it gives you a percentile that you're in. So if you have a low score, you're in this very low, you're like 0 to 20 percentile. But then if you're in these higher values, you're in the 80 to 100 percentile, and they give these kind of qualitative categories for it, right? So for example, if you have 600 parts per million of permanganate oxidizable carbon, that puts you right on that line between medium and high score. So you're at 60 percentile. Then you take, you do this for multiple indicators, and you take them and you combine them into this overall soil health score, right? So you have a couple physical ones, chemical ones, biological indicators, and you combine those together. Now, different people kind of combine them a little differently. Some people just take the average. Others do kind of like a weighted average. Some people then run it through these kind of logical tests. But the end result is that you get this single soil health score, ranging from usually 0 to 100. So you use that soil health score then, which is kind of the integration of all of your soil health assessments, to say, hey, here's the expected outcome, and you can then, change what you're doing accordingly. But, there's some kind of gaps currently, some things that we're still trying to grapple with here. So as I just kind of demonstrated a little bit, is the calculation of a soil health score is a little complicated, right? There's lots of steps, and it makes it difficult to really use on the fly. Another big kind of open question is, the applicability of these scoring curves across soil properties. As I kind of said before, you know, comparing a arid soil of the West with a Midwest small soil is going to be not a fair comparison. So the question is, how do we group these? What are the buckets that we put them in order to fairly compare things? So another problem is that this soil health score assumes the universality of some sort of desirable soil health property. For example, I did my master's work in California, and then I did my PhD work in the Midwest. So in California, you know, they often talk about, we want to retain water. What is your water holding capacity? And that would be something that matters a lot to them and should be heavily weighted in their soil health score. Whereas you go to the Midwest, they have the opposite problem where they have so much water. So what this means is you actually don't necessarily want a high water holding capacity, because then you're going to just have saturated soils and it's going to get flooded and all that kind of stuff. So there are kind of these nuances that we have yet to figure out and in calculating these soil health scores. Lastly, and importantly, I think, is that a lot of the work so far has been done on research farms. So those are not necessarily representative of kind of like the dynamic conditions of on-farm. You know, on-farm you have multiple managements kind of stacked on one another. So it's complicated to disentangle exactly what's going on sometimes. So this is kind of something that we need to work on moving forward. And this really is kind of the million-dollar question, if you will, that can we use these soil health metrics, these indicators, to successfully link management to a specific outcome? And so I think I wanted to start with an example that I think kind of demonstrates why we can't always assume that our theory is correct. So soil carbon is obviously central to a lot of soil health concepts. And generally, we think that more soil carbon is more productivity. I wanted to highlight two recent studies that Emily Oldfield did. I think they really wonderfully demonstrate that we can't always just assume that more is better. So the first one is a study they did using a high organic matter soil. So it was a little bit about 10% organic matter or 5% carbon. And what they did is they then diluted it down and looked at the productivity of wheat in this case, or the above ground biomass of the wheat in this greenhouse experiment. So they got this range of organic matter, all of similar quality, and they found that productivity kind of peaks around maybe in the 2.5%, 3% range. Now there are some additional kind of questions they layered in here about irrigation and nitrogen addition, but across these you can kind of see that productivity plateaus or peaks even in this neighborhood of 2.5%, 3%. Now, of course, that's one soil that they directly manipulated, but they also looked at this in terms of a kind of global meta-analysis. And this is maize yields or corn yields here across the world. And what they found is a similar point, around 2%, they found that productivity kind of plateaus. And so we see this kind of convergence between the direct manipulation and the meta regression that they did. And it shows us that this assumption that more soil carbon is better is not always true. And maybe we should rethink these assumptions and soil health indicators, those scoring curves, that more is better or more is worse. So when it comes to soil carbon, I think this dilemma comes up that I like how Henry Jansen phrased it. He put a nice point on it. He says, soil carbon, shall we hoard it or shall we use it? So hoarding it would be for things like structural soil carbon. Those increase aggregation, increase water holding capacity, right? Or we can use it. We can oxidize it, use it for soil fertility. Now, those two things are kind of directly in tension with one another. And so we need to be clear about what our desired outcomes are and which one we're managing for. Now, soil carbon is kind of slow to change over time. It takes, 5, 10, upwards of 10 years sometimes for the needle to move, substantially and for us to see management effects. Now, as a land manager, you don't necessarily want to think on the 10-year scale. You want to know if a new thing you're trying out is working on the 2-year scale, 3-year scale. So this was a study that I did with some colleagues to say, can we use these soil health metrics, some of these common soil health metrics, to tell us which direction we're headed? So to do that, we had a total of 76 sites spread across 10 states, about 1,100 samples. It was a combination of researched plots and on-farm work. And what we did is at each of these sites, they had some sort of paired management comparison. So for example, conventional tillage versus no tillage, annual versus perennial systems, organic versus conventional management, the use of cover crops or not using cover crops, and variations in rotation. And I did want to point out here that kind of all 4 of those soil health principles are represented here. So we got a good range of soil health building practices and sites represented here. So what we found was actually really interesting. So what we found is for each site, if we kind of put them together in a basic regression where we have on one axis mineralizable carbon or respiration, and on the other axis this permanganate oxidizable carbon, we found an interesting trend that popped out across all of our sites. So for example, these positive residuals, so a management that tends to stabilize organic carbon, those points all end up being above the line, whereas things that are using it are below the line, right? So this is the tension between hoarding it and using it. And we found it, so for example, in a couple different sites here, we had tillage comparisons. So this is one in Michigan, and we found no-till was consistently above the line, but regular tillage was below the line. Found the same thing in Maryland, and the same thing, this is a California site. So what we did was we saw similar things for things like compost addition, manure, use of cover crops, biochar, et cetera, et cetera. So the way we interpreted this is that when you are trying out managements that more permanganate oxidizable carbon relative to mineralizable carbon indicates greater stabilization, that hoarding of organic matter, whereas greater mineralizable carbon relative to the permanganate oxidizable carbon represents greater mineralization, right? So what does this mean? What it means is this is a kind of quick way of assessing which direction you're headed. So far, it's just associative. We're looking to follow it up, but it is a framework that is potentially usable by farmers and land managers. You can go out in a field and say, I'm going to try out cover crops. So half your field, you put in cover crops, half you don't. And you go out, take the samples, and you say, hey, are cover crops giving me the thing that I'm looking for, whether that's organic matter building or organic matter using. So anyways, it makes for an easy comparison that is pretty usable for each person. But the real big question is kind of centered around productivity. So, this is something that is often claimed by proponents of soil health, but it's not super well reflected in the literature. So the question here is, do biologically active soils, do healthy soils produce more yields than less active or less healthy soils? So this was some work that I did in kind of the Eastern Corn Belt during my PhD, and we had a couple different states represented here, and we wanted to constrain this to rain-fed corn systems. Rain-fed systems are a little bit different from irrigated systems, so we wanted to make sure to have a fair comparison here. And we used replicated nitrogen rate trials. So we wanted a minimum of 3 nitrogen rates, and then also a check plot or a true control where they got no nitrogen. We ended up with about 30 sites representing a combination of on-farm and research trials. And across all of these sites, we had these soil health practices kind of layered on. So we had N rates, but then also there were these practices that build soil health. So things like cover crops. And we had a bunch of different cover crops. So things like barley, rye, radish, and clover. Then we also had a couple different types of tillage, chisel tillage, moldboard, and no-till. We also had a bunch of different rotation lengths, rotation complexities. So everything from continuous corn to a couple corn, soybean centered rotations. So corn, soy, corn, soy, and corn, soy, as well as things like corn, soy, wheat, and corn, wheat. We also had manure on there as well at some sites. So these all kind of layered in to give us a big range of potential soil health values, if you will. Now, we were not, somehow excluded from grappling with these problems that I mentioned earlier that these soil health scores have to confront. So we still have to think about the fact that each field is going to have a different starting place in terms of their overall soil health, right? And that soil health is not something that we can go out and directly measure. And again, we're going to need to measure multiple things to get a good handle on soil health. So we kind of took a stab at doing this, and I want to show exactly how we did this. So the first one is addressing that different kind of baseline soil health. We use something called multi-level regression. Now, everyone stay with me. Hopefully a little bit of explanation of statistics won't cause you to tune out here. So what often happens with multi-site studies is we have several different sites. So here, each color is a different site. And then we have a couple different observations within that site. We take a couple different samples, right? So that's each of these dots here. So once we have all that data, there's kind of a couple ways that we can deal with it. So the first one is something called complete pooling, where we take every single observation and we throw them together on this type of graph. And what often happens is it gives us this relationship, right? But we can see within that each site kind of clusters by itself, right? So here we have the purple observations are kind of close together, and the red ones are here, and the yellow ones are here. And that's helpful if you're taking this kind of very zoomed out view of soil health and you want to look at it across, a region or something like that. But if you're the manager of this red field, you want to know is, how does my soil health, do improvements in soil health relate to productivity within that field? Now, another option is that people just average across all the fields, or all the observations within a field, I'm sorry. And what that does is that just gives you an average soil health per field. This is no pooling. So that will underestimate your effective soil health. So we need to figure out a way to kind of balance that, and multi-level regression lets us do that. So what happens is when we have those kind of 5 different fields with a couple observations from each, what we can do is we can let the starting place, that intercept, vary for each field. So then what we do is we're looking at soil health and we say, is the effect of soil health on yield consistent across fields, right? Relative yield varies from field to field. Some fields are just more productive than others. But as we increase soil health, do we have a consistent change in yield there? Okay, so that's the first piece we wanted to think about. Up next, we still need to figure out a way to measure soil health and have multiple variables, right? So we can't directly measure it, and we need to figure out how to combine these and blend multiple measurements together. Now, to do this, we use something called latent variable analysis. So what latent variable analysis does is it says these different measurements are correlated with one another, and that those correlations, that shared information, is meaningful. It describes some underlying trait or process. That underlying trait or process is referred to as the latent variable. Now, latent variables were developed for, the quintessential example is intelligence. So intelligence is not something that we can go out and directly measure. So instead, we kind of blend together a bunch of different measurements. So for example, it might be your math test score, your reading test score, your ability to solve a Rubik's Cube, how quickly you can put Legos together. Those are all maybe things that come together, and each of them imperfectly represents intelligence, but together they kind of give us some picture of intelligence. Now, similarly, we can think about soil health the same way, right? Soil health is something that's there and it's changing, but none of our indicators quite give us the full picture. So we used 4 of these soil health indicators. So again, permanganate oxidizable carbon, mineralizable carbon, soil carbon, and soil protein. So we used those 4, and we did this process to say, does each of them need to be there? Do we really need to measure all 4? And we found that mineralizable carbon actually doesn't provide us any unique information about soil health when we're using these other measurements. So then we needed to kind of put all of these pieces together in an actual testable model, right? So we started with that soil health variable that I told you about, that blend of all 3 of these measurements. And then when we're talking about nitrogen, we also want to think about your pre-plant nitrate, right? How much nitrate is there at the beginning of the season? And also you want to think about just how much nitrogen fertilizer you're adding. Now, all 3 of these things kind of come together to inform your overall yield. But those kind of 3 components here, pre-plant nitrate, soil health, and nitrogen, can all have interactions with one another as well. So we wanted to account for those as well. Then we actually run this and figure out what are the main effects here. And we found, first off, that pre-plant nitrate wasn't actually that strongly related to yields. That kind of fell out of the model and it wasn't that strong of an effect. But we did find that increases in soil health, that composite blended variable here, related to increases in yields. Now, it's hard to get an exact number on this, given this is just one study, but we were looking at about a 10% average increase in soil health related to about a 5% average increase in yields. So that's a good information to have there. Now, we also found that nitrogen was increasing soil health. So N fertilization is needed for soil health. And it was in fact building it within reason. So what are the implications here? So first off, we were able to kind of test and verify that in fact, healthy soils are more productive than unhealthy soils. What does this actually mean in terms of management, right? So what it means is if you are investing in something like soil health and you're using these different practices, you have some cushion for your N applications. So if you're going out and you're about to apply your nitrogen fertilizer and you say, should I add that extra 20, 50 pounds, however much it might be, as an insurance policy just to make sure you don't get a yield hit? Well, if you're investing in soil health and you have some of these practices in place and your soil health indicators are going up, it should say that you have some cushion and you can maybe just apply that little bit less and you can have some confidence that you won't get that yield hit. Think of it as an insurance policy. So kind of the overarching thing I want to impress upon you and talk about with these soil health indicators is that this is far from settled, right? This is all very new and we're still figuring things out. But as a reminder, the kind of take homes currently, first off, soil health indicators are not an end to themselves, right? We don't have a lot of confidence yet to say that these indicators are representing X, Y, or Z component of soil health, some facet of soil health. So therefore, you should use other metrics to also triangulate, right? Don't just rely on soil health indicators, but they are a good piece of the puzzle. And for that reason, don't just stop here. We want to make sure that we're connecting to outcomes. Up next, when you are implementing a management and you are assessing what that management is doing with soil health indicators, you want to make sure that you're measuring the signal, right? Differentiate the signal from the noise. So how to get around that, how to do that. So first off is replication. Run the replicates a couple times so that way you have a good idea of how much of it is the noise and how much of it is the signal. Also, since we do have some variability from lab to lab and also in space and time, you should be consistent in how you sample and where you send your samples. Lastly, be careful with how you interpret these soil health indicators, right? We're still figuring this out and making sense of them and figuring out how each of these represent some process, some biological process. So I would say for right now, a good idea is to don't bet the farm on it, literally or figuratively. Just be sure to take it with a grain of salt, and but an optimistic grain of salt, you know, because I do think that there is some value here. So with that, I just wanted to say thank you, and I think we'll have a couple minutes now for questions.
Thank you very much for sharing your perspective with us, Dr. Wade. That was some very great information. So now we have a chance for questions. So many questions have already been submitted. So we'll start out with one from Dr. Clute. This one comes in from Drew. Could you say a little bit more about the limitations of the Solvita paddles? What indicators might help growers to know that their soil is not well suited to Solvita due to having high respiration before they commit to that lab or method? Or is it simply trial and error?
Good question. I wouldn't be able to give you a definitive answer. Let me just turn this light off here. I would say one of the things that Solvita has done in any case is that I should have mentioned this in the talk, is that I think they've already addressed that and given you a lot more headspace with those Solvita panels. So you may want to check it out with them, because obviously, you know, if you can do those numbers in-house, it's pretty good. But I would say that once you're getting to soil, if you're getting to a 24-hour respiration of over 500, you'd want to check that out. And so one of the things you could do is possibly kind of do a range where you know you've got low versus a much higher soil test, CO2, and do it with your Solvita panels in your own lab, and then have it checked out by you know, guys like Rick Haney or the guys at Ward Labs or other guys who actually have a Franken breather, who can breathe a lot, you know, who can actually measure a lot more carbon. What we just noticed initially, so apologies to the Solvita guys. I think they have certainly addressed this more recently, but we did initially notice that 100 parts per million of CO2C was somewhere where we saturated out. And I think that has been addressed to some extent. To what extent, I don't know. But once you're getting to 800 or 1000 ppm, you may want to look for a different method.
Great, thank you. Dr. Wade, would you like to add anything to that before I have a question for you?
Just that I have experienced the same thing with Solvita paddles. I haven't used them in the last couple years, but I just want to back Buzz up on that. Same.
Great, thank you. And again, Buzz, feel free to jump in as well after this question for Dr. Wade. This one's from Stephen. As an additional source of variation, should we be thinking about variability within a field? I'm assuming that's in space, but probably also time. And what is the right sampling scheme for detecting a signal from the measurements?
Yeah, this is a great question. So we should definitely be thinking about it. One of the reasons that I kind of didn't discuss it is I haven't seen much on that. I've seen a little bit where they compared soil health indicators to your regular agronomic tests, a malic extraction, et cetera. They found that there wasn't a lot, this spatiotemporal variability in space and time there was about the same between your nutrient tester and your soil health tests. So there's no reason that you need to do, say, denser spatial sampling for these soil health tests. It was only 3 fields, unfortunately, because it does take a lot of labor to do this. But, you know, people are starting to look at it, and it's a source of variability. We just don't know a whole lot about it yet. Or I should say, I don't.
I think one of the things, you know, it's a great question. And, you know, I can walk 40 foot in a field and it's going to change. And so I do a little bit of consulting. And what we've basically brought it down to is, because we know that we can't always get a representative sample of, say, a 20-acre field each year. You know, you're not going to be able to repeat it exactly. So what we've resorted is to basically soil health points, or soil sample points. And we basically GPS points, and, you know, we GPS them at a density of 1 every 10 acres, 1 every 5 acres, 1 every 20 acres, depending what the producer wants. And we start looking at common topography and common soil series. So, you know, and we've been able to go back and really find great relationships where we're just taking a sample at a particular point. As soon as you start taking a composite, you know, you're running into a danger of the average. Or either way, you're going to run into averages. So.
Great, thank you. Yes, the average versus target is definitely a factor. For Dr. Wade from Gary, do most labs carry out biological tests? And again, Buzz, you may be able to speak to that too.
So most labs currently do not offer most of these biological tests. The most common ones right now are the mineralizable carbon or the respiration. Most of them use an erga, like I think Frankenbreather style, as Buzz called it. So more labs are starting to offer that, but, you know, since we're, as researchers, still trying to get our arms around this, and as I mentioned, trying to get this in a form that is high throughput, most labs aren't offering it yet. But there are some, like Ward Labs, for example, that are starting to offer it. And also university labs like Cornell, or plug for University of Missouri's soil test lab here too. They're offering a couple of these as well.
Wade, what about the Illinois soil nitrogen test? Are they doing that in their labs as well?
Some places offer the ISNT and others don't. Yeah, it varies from place to place. I think according to the demand. I think Ward does it, though, does do the ISNT, but I can't recall 100%. I don't use the ISNT very often myself, so don't quote me on that.
Great, thank you. And I think here in Washington State, at least, it's pretty easy to just call up the soil test labs and ask them if they do that the particular test that you're looking for. I know here in Moses Lake, they have Soil Test Farm Consultants offers Solvita test from a regional standpoint here. Here's the question for Buzz from John. Why not just spoon feed the growing plant what it is requesting via plant sap analysis? Can you speak to that?
Well, yeah, you're probably dealing with high value plants. So certainly if you can do that and you can afford to run across it with, you know, the foliar spray or whatever you're using, certainly that's a good thing. But if you're in a commodity like corn and soybeans, that's a little bit tougher to do. But certainly that's, you know, using plant sap analysis is good. I have very little practical experience of that. But I know, you know, guys like John Kempf have talked about it, and they offer some very, very good services. So I'm not the expert on that, but if you're going to try that on corn, soybeans, you're going to lose your shirt, I think.
Great. Thank you. For Dr. Wade from Gary, are biologically active equals organic soils?
I'm not entirely sure what you mean there, Gary. Sorry. If you're talking about high organic matter soils, meaning like a histosol, a muck soil, those are somewhat biologically active. They're pretty active, although it will depend on if they're drained and redox status, etc. In terms of organic soils would have a lot of biological activity if you let them.
Great, thank you. Also, from Thomas, you seem to oppose stabilization to mineralization. This is for Dr. Wade. Isn't it possible to achieve both? Using a bank account approach, can't we increase our savings while compounding our interests?
Yeah, that's an interesting question. I don't want to extend an analogy past where it should go, extend a metaphor rather. I think stabilization and mineralization on aggregate are in tension with one another. I mean, mineralization is literally CO2 coming out of the soil. So those often are in tension with one another. Of course, we're trying to balance the two, and so they don't have to be in opposition. They can also be in balance. Now, if we're talking about other nutrients outside of carbon, I do think there's possibility for nutrient cycling, et cetera. If that's what you're trying to get at. But in terms of carbon, I think stabilization and mineralization are, in fact, opposing processes most of the time.
Buzz, is there anything you would like to add to that?
Well, yeah, I wouldn't really say they're opposing. I would say, Jordan, would you agree that they're more complementary processes?
Yeah, I mean, I would agree that they are complementary in that you want to have them both for sure.
Correct. Yeah, and if you have a look at the Haney analysis, they often look at the WEOC to WEON, and then the CO2 to WEOC ratio. So they also have a measure for that. So that's certainly, I haven't had that much experience. So I'm really glad you gave your talk. I heard you talk, Jordan. So those are things I think we need to just understand, that natural systems are in tension. But I would say that, yeah, as we grow that carbon base, we could probably also learn to use it a little bit more as we go along. So yeah, it's a natural system. So everything, it's no longer formulaic anymore. It's a system. So that's where it gets tricky.
Systems work, but it's what keeps it interesting. How is mineralizable carbon determined? Solvita, combustion. This is from Stephen to Dr. Wade. But again, Buzz, you're welcome to chime in as you like.
Yeah, that's a good question. Methodology matters on a lot of these things. For the studies that I was talking about, we used the infrared gas analyzer. So that's just measuring CO2 by, the respirometer. So that, as far as I'm concerned, that's kind of like the gold standard in terms of CO2 loss. It's a little expensive for that equipment. A lot of people don't have it. So, that's where Solvita comes in, and I think where they shine, especially as we mentioned before, at lower CO2 rates.
Great. Here's one from Gary. Doesn't soil health include nutrient soil levels? I'm not sure who that was directed towards, so I'll let you guys decide.
Buzz, go for it.
Yeah, that's also a great question, and I think I just loved that picture that Jordan had, with the obstacle course and everything. It just depends where you are. So I have seen soils that have, according to our land-grant university, just, dangerously low phosphorus levels, and you go out and you look at the plant and the plant has no phosphorus shortage. So that's a movable feast. What we've seen in some of our soil health fields, these are, because I'm not from a land grant university, I work with guys who have done this for 7 or 8 years, is we've slowly seen our soil test potassium levels seem to have stayed steady. Our soil test P levels have come down, yet we don't see soil test P deficiencies. And this probably, if you go to guys like Gabe Brown and his name escapes me, in South Dakota, oh gosh, I can't remember his name. We're also seeing something there. So again, remember, a soil test value is a value that is, we take some kind of chemical, whether it's a nitric and sulfuric acid, or what is the Morgan one, that's ammonium something or other. So we're kind of using chemical methods to kind of look into the soil, and as Rick Haney says, we're trying to, we're trying to interrogate the soil and wring an answer out of it, whereas in nature, those minerals come out much more easily. The other thing, and then I'll stop here, is that a lot of times, especially with something like carbon, phosphorus is, once you've got mycorrhizal relationships going, with things like phosphorus and other minerals, it almost seems to be what some of the studies that we've seen is our plant mineral content is almost or it is independent of what we're finding in soil test minerals.
Well, I guess I will add to that as well, that to me, when I think about soil health, the novelty that soil health brings, and the thing that it really tries to lean on, as you may have gathered from both our presentations right now and previous presentations, is that we're trying to incorporate biological components and biologically cycling nutrients. So I think when we're talking about that, then it's going to depend on the nutrient of interest, right? So obviously nitrogen is biologically cycling, phosphorus biologically cycling, sulfur to a lesser degree. But, when you look at things like potassium, that's less of a biologically cycling thing. It's more just your basic chemistry and your kinetics there. And this is where soil tests are trying to mimic that. But, as Buzz said, there are imperfect measurements, so we're just trying to look at it at this broad scale. But when it comes to soil nutrients, I think it's going to depend on the nutrient of interest a little bit.
It's too bad that some of the enzyme assays are so cumbersome to undertake in the lab, I think. Okay, for both of you, from Russell, if yields peak at 5% organic matter and 2 to 3% carbon, can we increase yield and those numbers by adding biologicals, such as bacillus and fungi species?
Yeah, I guess I can go for this. So that relationship is an observational relationship, right? That it peaks there. And I think there's a lot of things that we're still trying to tease apart for why that happens. But to your question about increasing yields with biologicals, I think it's mixed results so far. It'll depend on the biological. I think they were talking about this yesterday, I believe, or maybe it was earlier today. I don't know. Time is blending together. But anyways, moving the needle in terms of which microbes are there is really difficult to do with biologicals. A lot of them are already there, and you just need to make a good home for them, right? You need to build aggregates, et cetera. They're already there. You just got to lean into the good stuff. You don't need to be necessarily adding things as much. And what you're adding such a tiny amount of biologicals that what's there is probably going to be both good at competing and just more of it.
Yeah, I think I would agree with Jordan, especially, if we're trying to manufacture biologicals in a lab, you might have a very non-diverse set of, say, mycorrhizal fungi, and they say, oh, we've got all these spores. But it's kind of like dropping a bunch of Marines into Afghanistan and then not resupplying them, right? So your Marines, all your infrastructure is there. You've just got to provide that habitat of food and water and a place to raise their young. So that's exactly like above ground habitat. You're doing that. The other thing is possibly, the emphasis that we've seen in the last 7 years of soil health research with farmers is that our yields have pretty much stayed the same, but our costs of production have just gone down and down and down. And the guys that I work with are staying in business, and the guys who are going for the big yields have gone out of business. So it's as simple as that, where I think sometimes, industry is attracting us with bigger yields, spray azapazine to kill, whatever, some kind of nematode, and you kill everything else. And it might be effective for a single year. But what we found overall is our risk of production has gone down simply because our inputs may be between $100 and $200 an acre less for, a broad acre farming.
Great. Thank you both for sharing on that. I do want to acknowledge that we are right at the 2:30 time point, but we just have a couple more questions. So I do invite anyone who has to be elsewhere to go ahead and take off. Please fill out the poll before you leave. But again, for those who are interested, let's stay on for just another couple minutes while we wrap up these last couple questions. Okay. For both of you from the hangar, when is the right soil sampling time for biological tests for soil health? Careful, I'm going to call on one of you.
All right. Yeah. So that's another good question. I'll tell you, our experience is probably the best time for a biological soil test is just before planting. We have found, for instance, with PLFAs and also the Haney test, that, and this was where we compared cover crop versus no cover crop, we found significant differences just before we planted. And we found zero difference at the end. So if you say taking a sample sort of after you've harvested, and this is often the habit of a lot of broadacre farmers, we'll take a sample after we've harvested, then we're going to add our whatever. You've got a whole winter going through either fallow or with cover crops where your soils are going to change a lot. So my recommendation would be as close to planting as possible. I would also say that's when your soils are often most biologically active.
Yeah, I would agree with that, that the closest you can get to planting is the best, obviously. Now, there are lots of time demands when you're planting, et cetera, especially in annual systems. You're waiting to get in, and, at least in the Midwest, et cetera, it can get really mucky, and you got a 24, 36-hour window to plant. So, depending on which things you're getting measured, some of them are more sensitive than others. For example, Buzz was talking about PLFA. That one is more sensitive. So if you're measuring that, you'll get the best bang for your buck doing it in the spring pre-plant. But, something like the soil protein assay I was talking about, it's a little bit less finicky, and it's a little bit harder to move that one. So you could probably get away with sampling that one around harvest, if you will. When it comes to like perennial systems, I think this is actually an area that is really ripe for research. If you have a tree crop or any sort of perennial thing, vineyards, et cetera, I think it'll be, that'll be an interesting question. Really, the thing that we're trying to get at is, when can we sample to get the most reliable information about what is coming in the season ahead, or 2 seasons ahead, et cetera. So that's a hard question to answer, especially if we don't have the outcome of interest already. So that's a thing to keep in mind.
If I can briefly add to that, from what I've heard with soil sampling guidelines in general, if you are wanting to compare between years, the most important thing is to keep your sampling time consistent between what you want to compare to. Thank you both for sharing your insight on that. Okay, we have 2 more questions. One from Qian Yi. Could you talk a bit more about the more soil carbon equals more production part? What factor causes the decline of yield and biomass in high organic matter?
Well, since I was the one who opened that can of worms, I guess I'll take it first. So the short answer is no. I would be guessing if I said something. Previously, if you had asked me, I would have said that high organic matter soils are generally just wetter, they're in colder climates, et cetera. If you're getting up to 8, 9, 10% organic matter, that's really high organic matter. And but anyways, those studies that I highlighted really kind of changed my mind on that. So I'm not really sure why it's happening yet. They would probably, the studies authors would probably be a better source for that. And I'm sure they're doing follow-up work on it. They're really chasing it. So I would punt it to them.
Great, thank you. Our last question is, looking ahead, where might we be in 10 years? And I'd love to hear both of you chime in on this one.
Well, since I'm the older guy, I'll go first. So, I think what industry has taught us to do is to go out and look for silver bullets. You add this biological, you add this chemical, you add these micronutrients, and it'll solve all your problems. And really what we're doing is, as we go down the road, is we see this system symmetry. And you go to guys like Brendan Rocky, who's a potato farmer, and he can actually give you the progression of his system over time, where he started, with a single species cover crop, and then he added a multi-species cover crop, and he said that's where he saw the biggest change. And then he started intercropping multi-species in his potatoes, and then he started adding animals. So to my mind, I think we're looking at systems. And the thing that really kind of, am I allowed to use a cuss word? Okay. The thing that really ticks scientists off is that systems are really hard to measure. If you're just measuring a single variable, you can go and publish a paper. But when you're stacking all of these things on top of each other, then you're finding that 1 + 1 + 1 = 7. And it's like, what's going on? So I think, as we go along, I'm sorry, I couldn't use the cuss word, but as we go along, I think we're looking in the long run at stacking systems. And my other thing is not only stacking systems, but stacking enterprises. So, we're working with ranchers in South Dakota that are not only ranching and providing, livestock, but they've got a quail hunting operation. Some of them have eggs, some of them have chickens, and so there's a lot of stacking that's going on. And so we're able to feed more people on less land and employ more people on less land. And that just excites the heck out of me in terms of soil health. For us scientists, it kind of presents a bigger problem, but it's a lot more exciting. And it moves us away from being dependent on, larger and larger industries, and starts to make food going a little bit more local.
Well, that was great. That's hard to follow. I guess when I think, where we're going to be, I personally like to think about three kind of different things. Is one, I think we need to keep developing indicators, right? We have a set and we're testing them, et cetera, but we might need to add some more or subtract some, who knows? But I think we need to keep working on that. And that's, lots of hard work being done on that. So I'm optimistic about that. The second thing is that I want to get away from the "it depends." Obviously, that is true, but I think as scientists, we should ask a little bit more of ourselves to really kind of tease it out and give meaningful answers and reliable answers, especially in the realm of soil health. And the third thing is that I like to think about, and this is part of the "it depends," but I want to think about kind of soils. We have lots of soil pedology that has been done, lots of soil classifying, et cetera. And I think that that's kind of undervalued oftentimes. And we talk about it, we kind of wipe it aside and we say, oh, it's a high clay soil, or it's a this soil, but there's so much more that's there. And I think that we can start to incorporate that into soil health and soil fertility, kind of thinking more broadly. I think that that will be a huge breakthrough. And I think that we can make a lot of progress in that realm. So those are the three things for me in the next 5, 10, it might even take longer, maybe 15, 20 years, who knows? But those are the things that I'm looking at.
With those great answers on more dialed in answers from scientists and function stacking in our systems and synergies, I really want to thank you both for being here today. It's been a real pleasure to work with you. I also want to thank the Washington SoilCon team for putting on this excellent event. It's a real, as much as we all love the Zoom environment, there is some real benefits in allowing us all to gather in this space and cross-pollinate despite being in different places. So with that, audience members, please go ahead and fill out the poll so that this wonderful SoilCon team gets your feedback on the event. And with that, thanks so much for joining us today. Hope to see you again tomorrow morning. Thanks.
Thank you so much, everyone.